Hello friends, let's revise microbiology quickly. So first point, we have this hepatitis B virus, genome of hepatitis B virus. P gene, it codes for DNA polymerase. Pre S1 for L means large protein. Pre S2 is M protein, means middle protein. What is C gene? That codes for hepatitis C antigen, and pre-C region HBe antigen. Next point, Bamble disease. What is this? Also known as Bornholm disease, or this is caused by Coxsackie B virus. Next one, we see this pigeon and bat and their droppings. So this is histoplasmosis, cause of histoplasmosis. Droppings may contain this fungus histoplasma that contaminates the soil. So through the inhalation of such spores, we may get this disease, histoplasmosis, also known as cave disease, Darling disease, or Ohio Valley disease. This is endemic in USA. It is thermally dimorphic fungus, means on two different temperature it has two different form. So mycelial form, that is at 25 degrees Celsius, that is in the soil, and yeast form, that is tissue, this is at 37 degrees Celsius. Yeast form is at 37 degrees Celsius in tissue. This is common in AIDS patient, and the conditions this mimic like cold or flu, pneumonia. Next condition, what is this? This is the cetrimide. This is of course antibiotic, and this is acetyl trimethylammonium bromide. It is a surface active agent, and this act on phosphate group of a cell membrane. Next point, formaldehyde. This acts against amino group of proteins. Next, what is Treg cell? This is CD4 plus a T cell. This combination is Treg cell. Next, what is this? Barrel shaped fungus. So this is Coccidioides imitis. This is dimorphic fungi, means this occurs as a saprophyte in the soil, and the shape of this fungus in soil is just like barrel shaped, and in a host it occurs as a parasite, and it occurs as spherules and then endospore in host. Next one, this some different name of these conditions. So histoplasmosis, cave disease, Darling disease, Ohio Valley disease, and reticuloendotheliosis. All of them are same. Coccidioidomycosis. It's also known as valley fever disease, desert fever, San Jacqueline Valley fever. Blastomycosis. It is also known as Gilchrist disease, Northern American blastomycosis. The next one is paracoccidioidomycosis. This is also known as Brazilian blastomycosis and South American blastomycosis. Next point, this is quorum sensing. It is the regulation of gene expression in prokaryotes. This is cell to cell communication, that's in response to fluctuation of cell population density. So what the gene will do in when the cell population is changing, that is the quorum sensing in prokaryotes. Next point is this is the difference between MHC1 class and MHC2 class. So MHC1, this is the peptide binding groove that is between 1 and 2. How we can remember? When we write 1, we can write this like, okay, so here is the point 1, here is point 2, and here is point 3. Okay, so all in series. So this is so in peptide presentation is in this groove, alpha 1, alpha 2, and this is presented to CD8 T cell. Okay, and this system of presentation, MHC class 1, is present on all nucleated cell except sperm, and it works for only for endogenous peptide only. And here CD8 cells bind with cytotoxic T cells. What about MHC2? So here is the groove, just like 2 means 2 different hand. Here is the alpha 1 and here is beta 1. Alpha 1 and beta 1, that 2 hand is required to catch or peptide of exogenous antigen, that is peptide. And exo means external is foreign, so foreign is 4 CD4 Th cell, so that's bind with CD4 Th cell, T helper cell. And this is the endocytic pathway presentation of the peptide. Okay, this present in antigen presenting cell. So again the mnemonic is here. What is 2 hand is required to hold foreign peptide, so alpha 1, beta 1, and foreign peptide, so CD4 has the role. Next one is the differences between Strongyloides and Trachinella. So Strongyloides is the ovoviviparous, means it lay eggs, but egg has fully formed larva that hatch out immediately. And this is the Trachinella, this is viviparous, this directly produces larva. And other worms that can produce a larva is Vauchereria and Brugia. Next and the last topic for today's this small session is Serenity test. What is this? This is used for detection of this was used and this is now obsolete. This is intro-invasive E. coli and Shigella. This test is actually animal pathogenicity to produce disease in animals. So what we are doing, we take the sample, possible pathogenic sample, and inoculate that one to the eye of a guinea pig. If this develop mucopurulent conjunctivitis or keratitis, then Serenity test can be said as positive. So this test is now not performed. So friend, thank you. Please like and subscribe my channel.